Cellular division is an essential process for the continuation of all life. Cellular division is used by organisms to reproduce, grow, and repair. Mitosis is the process in which a cell splits into two identical daughter cells, and meiosis is the process in which it splits into four daughter cells with half the number of chromosomes, none of them in pairs. The first stage of mitosis is the interphase. Here, once the cell is grown, enzymes begin to unwind the DNA in the nucleus. As the double helix is straightened out, the nucleotides are separated and joined with free nucleotides. This doubles the amount of DNA, and so a chromosome with two chromatids is formed. As prophase begins, the DNA finishes forming chromatids, and enzymes break down the membrane surrounding the nucleus. Eventually, the chromatids are fully condensed, and the spindle, made from the cell's centrosomes, begins to form. By now, everything is ready to go. The spindle lines up the chromatids along the middle of the cell. Some spindles attach to the center of the chromatids, centromere, and others stretch across the whole cell. Now it gets exciting! The centromeres of the chromosomes divide to separate the two chromatids. The spindles attached to the centromeres then retract to move the separate pairs of chromosomes to either side of the cell, while other spindles elongate so the cell is pushed apart. The separate chromosomes now form two new nuclei, and a cell plate forms across the middle of the cell. Cytokinesis now begins. The cell starts to split. In this example, we see what would happen to an animal cell. The ends pinch in, creating a cleavage furrow which pinches in far enough to split the cell in two. And that's mitosis. This process is what is used by multicellular organisms to repair their bodies or to grow, and is used by unicellular organisms to reproduce. Meiosis is split into two stages, Meiosis I and Meiosis II. Naturally, we'll begin with Meiosis I. During Meiosis I, the stages of interphase and prophase are pretty much identical to that in mitosis. It is during metaphase where the differences begin. During metaphase, the bivalents, matching chromosomes in a pair, line up. This contrasts with mitosis's metaphase, in which the individual chromosomes line up, with each chromatid on either side of the cellular equator. By having the bivalents line up, genetic information can pass between the two chromosomes, and there is also a 50-50 chance that either chromosome will end up on either side, therefore shuffling the genetic information. Interestingly, in humans, this allows for over 8 million different combinations of genetic information to be passed to the daughter cells, and thus a couple have the genes to create over 70 trillion different children. The pairs of chromosomes are now separated, so now each side of the cell has half the number of chromosomes a cell normally would. The cell then divides, creating two daughter cells with half the number of chromosomes, but still having each chromosome present as two chromatids which would have been pulled apart in mitosis. Meiosis II is practically the same as mitosis, except that the cells only have half the chromosomes. During Meiosis II, the chromatids from Meiosis I are pulled apart, and so four daughter cells are ultimately created with half the number of chromosomes. And that's meiosis. This process is only used by sexually reproducing multicellular organisms, so that they are able to reproduce by having two gametes, eggs and sperm, fused to form a zygote. So to sum up, mitosis produces two identical cells by producing a copy of its genetic information present in chromatids, and then putting the copy and the original into two separate cells. Meiosis, on the other hand, produces four cells, in which two are identical to one another but not to the other two, by halving its genetic information into two cells, then replicating those into four.